Human DNA, the complex blueprint of life, is nothing short of extraordinary. If you were to stretch all the DNA in every cell of your body end to end, the length would extend far beyond the confines of the Earth, all the way to the Moon and back thousands of times. To put this into perspective, the human genome, the complete set of genetic instructions for a human being, consists of approximately 3 billion base pairs of nucleotides. These base pairs, which make up the DNA structure, are incredibly small, with each base pair measuring about 0.34 nanometers in length. This seemingly infinitesimal measure hides an astonishing fact: if you were to lay out all the DNA in your body, the combined length would stretch to about 10 billion miles. That's enough to reach the Moon more than 25,000 times. The sheer scale of this number gives us a profound appreciation for how deeply embedded our genetic code is within the very fabric of life. The DNA inside each human cell is not only vast but intricately packed. Every cell in the human body contains roughly 6 feet of DNA, but it is packed into a nucleus that is just a tiny fraction of the cell's size. This packing is achieved through a series of sophisticated folding mechanisms, which allow the long strands of DNA to be efficiently compressed without losing the ability to be read and replicated. The process begins with the DNA wrapping around proteins called histones, which then coil and fold into structures called nucleosomes. These nucleosomes further fold into higher-order structures, creating a dense, organized system that allows the entire genetic code to be stored in a microscopic space. Despite the incredible length of the DNA, it remains accessible for processes such as gene expression and DNA replication, where specific parts of the code are copied or activated as needed. This intricate organization ensures that the vast length of DNA in our cells can function as a highly efficient and organized database of genetic information. Understanding the vastness of human DNA is a challenge that has intrigued scientists for centuries. The human genome has been described as a blueprint of life, and in many ways, it functions as a set of instructions that dictates everything from our physical appearance to our susceptibility to disease. However, what is perhaps more fascinating than the sheer size of the genome is the way in which these instructions are decoded and expressed. While the human genome contains about 3 billion base pairs, only about 2% of this genetic material directly codes for proteins. The rest of the genome was once considered junk DNA, but recent research has revealed that it plays a significant role in regulating gene expression, maintaining chromosome structure, and even influencing evolution. In many ways, this non-coding DNA is just as essential to life as the coding sequences, as it helps determine when and where genes are activated or silenced. The discovery that much of our genome is involved in regulating and fine-tuning our biological processes has changed our understanding of genetics and deepened our appreciation for the complexity of life. The vast length of DNA is not just a curiosity of the human body, but an essential part of the way our cells function. One of the most remarkable features of DNA is its ability to replicate itself with incredible accuracy. Each time a cell divides, the DNA inside it must be copied so that each daughter cell has an identical set of genetic instructions. This process, known as DNA replication, is a highly coordinated event that involves a multitude of enzymes and other molecules working together to duplicate the genome. The DNA molecule unwinds, and the two strands are separated, with each strand serving as a template for creating a new complementary strand. Despite the enormous scale of the genome, DNA replication occurs with an error rate so low that the accuracy is near perfection. This precision ensures that genetic information is passed down faithfully from one generation to the next, maintaining the continuity of life. What's even more astonishing is the speed and efficiency with which DNA replication occurs. In human cells, DNA replication can take place at a rate of approximately 50 nucleotides per second, and the entire genome can be copied in just a few hours. This rapid process is critical for the proper functioning of cells, allowing tissues to grow, repair themselves, and replace damaged cells. Yet, even with its remarkable precision and speed, errors do occasionally occur. These errors, known as mutations, can sometimes lead to diseases such as cancer or genetic disorders. However, cells are equipped with a complex system of repair mechanisms that constantly monitor and correct these mistakes, ensuring that the genome remains intact over time. Despite the challenges of managing such a vast and intricate molecular structure, cells have evolved highly sophisticated systems to preserve the integrity of DNA, ensuring that it continues to function as the foundational instruction set for life. The vastness of human DNA also connects to our evolutionary history. The length and complexity of our genetic code are the result of millions of years of evolution, and our DNA carries with it the history of our species. If we compare the human genome to that of other species, we find striking similarities, but also key differences that set us apart. For instance, humans share approximately 98% of their DNA with chimpanzees, our closest living relatives. This close genetic relationship speaks to our shared evolutionary ancestry. However, the remaining 2% of the genome accounts for many of the traits that make us uniquely human, from our larger brains to our ability to speak and use complex tools. These small genetic differences, spread across our DNA, contribute to the emergence of human-specific characteristics that have allowed our species to dominate the planet. By studying the genomes of other species, scientists are uncovering the subtle genetic changes that have shaped our evolution and continue to influence our development. At the same time, understanding the scale and structure of DNA offers valuable insights into how our bodies interact with the environment. The environment has a profound impact on gene expression and can influence how genetic information is read and interpreted. Factors such as diet, stress, exposure to toxins, and even social interactions can all affect the way our genes function, often through mechanisms known as epigenetics. Epigenetic changes do not alter the underlying DNA sequence but can modify how genes are turned on or off, influencing everything from aging to disease susceptibility. This growing field of research has revealed the dynamic interplay between our genes and the environment, highlighting the complexity of the human genome and the many factors that contribute to our health and development. Despite the immense scale of human DNA, it is often the smallest details, such as individual genetic mutations, variations in gene expression, and environmental interactions, that have the most profound effects on health and behavior. The length of DNA is not just an abstract concept but a measure of the immense potential encoded within every cell. This potential is realized through the intricate processes that allow our genetic code to be read, interpreted, and acted upon by our cells, driving the biological machinery of life. As research into the human genome continues to advance, the depth of understanding about its structure and function only increases. Scientists are now able to sequence entire genomes quickly and affordably, making it possible to study the genetic underpinnings of a wide range of diseases and traits. The ongoing exploration of DNA's vastness and complexity promises to unlock new insights into everything from human development to personalized medicine. With each new discovery, we move closer to understanding the full potential of our DNA and the profound implications it holds for our future. The study of DNA, with its astonishing scale, continues to be one of the most exciting frontiers in science. As we continue to uncover the mysteries encoded in the billions of base pairs that make up our genetic code, we move closer to understanding not just the origins of life, but the very essence of what makes us human. Building upon the vast scale of DNA, we can delve deeper into the mechanisms by which genetic information is utilized and regulated. One of the most remarkable aspects of DNA is how it functions as a dynamic system, not a static archive of genetic code. Within every cell, the DNA is constantly being read, copied, and modified in response to the needs of the organism. This constant activity is what drives cellular processes such as growth, repair, and reproduction. The process by which genetic information is turned into functional molecules, like proteins, is known as gene expression. This involves two key steps: transcription and translation. Transcription is the process by which the DNA code is copied into a messenger RNA molecule. This molecule then travels from the nucleus, where the DNA resides, to the ribosomes in the cytoplasm, where proteins are synthesized. The ribosomes read the mRNA in sets of 3 bases, codons, and translate the genetic code into an amino acid sequence, which then folds into a functional protein. The efficiency and accuracy of this process are crucial for the proper functioning of cells and, by extension, the whole organism. However, gene expression is not a simple, one-way process. It is tightly controlled by a variety of regulatory mechanisms that ensure that the right genes are turned on at the right time. One of the most fascinating aspects of gene regulation is the role of epigenetics. Epigenetic modifications are chemical changes to the DNA or the proteins around it, such as histones, that affect how the genetic material is expressed without altering the underlying DNA sequence. These modifications can be influenced by environmental factors, such as diet, stress, or toxins, and can sometimes be passed down through generations. This suggests that our genetic code is not only a product of our inheritance but also a reflection of the environments and experiences our ancestors had. Epigenetics has opened up new areas of research in understanding how genes are regulated and how certain traits or diseases can be influenced by factors beyond our DNA sequence. For example, studies have shown that stress can cause chemical changes in the DNA of individuals, potentially affecting their children and even grandchildren. This intergenerational impact challenges the traditional view of inheritance, where only the DNA sequence itself is passed down. Another layer of complexity is added by the fact that not all of our DNA is actively used in gene expression. The human genome contains vast amounts of non-coding DNA, which does not directly code for proteins but has been shown to have important regulatory roles. This junk DNA, once dismissed as a byproduct of evolution, is now understood to be involved in regulating gene expression, maintaining chromosomal structure, and controlling cellular processes such as DNA repair and replication. In fact, some of this non-coding DNA is crucial for ensuring that the right genes are turned on or off at the right times, and disruptions in these regulatory regions can lead to diseases such as cancer. The discovery of the importance of non-coding DNA has revolutionized our understanding of genetics, making it clear that our genome is far more complex than previously thought. The human genome's size and complexity also have significant implications for personalized medicine. As we learn more about how genes interact with the environment and how small variations in our DNA can lead to different health outcomes, it becomes possible to tailor medical treatments to an individual's genetic profile. This is the essence of precision medicine, treatments that are specifically designed to work with the patient's unique genetic makeup. For example, certain cancers have been linked to specific genetic mutations, and treatments can be developed to target those mutations directly. Similarly, variations in genes related to drug metabolism can influence how a person responds to medications, meaning that a one-size-fits-all approach is not always ideal. The ability to sequence an individual's genome and identify these variations opens the door to more effective, personalized treatments that can improve outcomes and reduce side effects. At the same time, as we explore the relationship between genetic variation and disease, we are also uncovering the ways in which our genes interact with one another. The vast number of genes in the human genome are not isolated entities, they work together in networks, where the expression of one gene can influence the activity of others. This interconnectedness adds another layer of complexity to our understanding of biology. For example, the regulation of immune system genes is highly interdependent, meaning that mutations in one gene may have cascading effects on the entire immune system. Understanding how genes interact in these networks is key to identifying the root causes of complex diseases such as autoimmune disorders, diabetes, and cardiovascular disease. As we continue to decode the human genome, the application of gene editing technologies such as CRISPR-Cas9 is making it possible to directly modify DNA with unprecedented precision. This technology has revolutionized genetic research and holds tremendous potential for treating genetic disorders. By cutting out defective genes and replacing them with healthy ones, scientists could potentially cure genetic diseases that were previously untreatable. However, gene editing also raises important ethical questions. How far should we go in editing the human genome? What are the long-term implications of altering our DNA? These are questions that society will have to grapple with as gene editing technologies continue to advance. The ability to manipulate our genetic code has the potential to cure diseases, enhance human health, and perhaps even extend lifespan, but it also comes with risks that must be carefully considered. While gene editing holds promise, it is essential to remember that our genetic makeup is not just a sequence of DNA. Our genes do not operate in isolation from each other or from the environment. The interactions between genes, epigenetic modifications, and environmental influences create a dynamic system that is still not fully understood. As our knowledge deepens, it will be critical to continue exploring not only how DNA is structured and replicated but also how the environment shapes our genetic expression. This broader view of genetics will be key to unlocking new treatments and therapies that harness the full potential of the human genome. The expanding knowledge of DNA's inner workings is reshaping our understanding of life itself. From the tiny molecules that carry genetic information to the complex networks of genes that govern our biology, DNA serves as the foundation of all living organisms. As scientists continue to uncover the mysteries of the human genome, we are beginning to realize that this vast, intricate code holds the answers to some of the most profound questions about our existence. From disease prevention to personalized treatments, the potential applications of DNA research are virtually limitless. Understanding the true power and complexity of our genetic material will continue to be a driving force in the scientific world for decades to come. In a world where new genetic discoveries are being made every day, one thing is clear: the story of our DNA is far from over. As we continue to explore the vast, interconnected web of life embedded within our genetic code, we uncover more about the forces that shape us, both as individuals and as a species. This ongoing journey promises to deepen our understanding of what it means to be human and how we can use this knowledge to improve health, extend life, and perhaps even change the course of human evolution. As we continue to explore the complexities of human DNA, it becomes increasingly clear that the study of genetics is not only about understanding the code itself but also about investigating the interaction between genetic factors and disease. One of the most compelling areas of research is the study of genetic predisposition to various conditions. While our genes serve as a foundational blueprint, the expression of these genes, and how they contribute to our health, can be influenced by a range of factors, including lifestyle, environment, and even social determinants. Diseases like cancer, diabetes, and heart disease often have a strong genetic component, but the way these conditions manifest can depend heavily on external influences. For example, an individual may inherit a genetic predisposition for a certain condition, but whether the disease actually develops may depend on factors such as diet, exercise, and exposure to toxins. This realization has given rise to a more nuanced understanding of health, one that moves beyond simple genetic determinism and emphasizes the role of epigenetics, the study of how external factors can influence gene expression. The concept of epigenetics has opened up exciting possibilities for preventive medicine. Rather than simply treating diseases once they occur, epigenetic research is providing insight into how we might intervene before diseases develop. For example, studies have shown that certain environmental factors, such as diet and stress, can affect the expression of genes associated with cancer. By modifying these environmental influences, it may be possible to prevent or delay the onset of cancer in individuals who are genetically predisposed. This approach not only holds promise for improving outcomes for individuals but also suggests that public health interventions targeting environmental factors could play a significant role in reducing the prevalence of chronic diseases on a global scale. Another fascinating area of research is the study of genetic mutations and how they contribute to both common and rare diseases. Mutations are changes in the DNA sequence that can occur naturally over time or be triggered by external factors like radiation or chemicals. While many mutations are harmless, some can lead to serious health problems. For example, cystic fibrosis, a genetic disorder that affects the lungs and digestive system, is caused by a single mutation in the CFTR gene. Similarly, mutations in the BRCA1 and BRCA2 genes increase the risk of breast and ovarian cancers. These mutations are often inherited, but they can also occur spontaneously. The discovery of genetic mutations has paved the way for genetic screening, the process of testing individuals for specific mutations that may predispose them to certain conditions. Genetic screening can help identify individuals at high risk for diseases, allowing for early intervention and tailored treatments. As genetic research continues to advance, the potential for personalized medicine is becoming more and more apparent. Personalized medicine involves tailoring medical treatments to an individual's genetic profile, allowing for more effective and less invasive therapies. This approach contrasts with the traditional, one-size-fits-all method of treatment, which often does not account for genetic variations between individuals. For instance, genetic testing can help doctors determine which medications are most likely to work for a patient based on their unique genetic makeup. This is particularly important in the treatment of conditions like cancer, where certain genetic mutations can affect how a tumor responds to specific therapies. By understanding the genetic basis of a disease, doctors can choose the most appropriate treatment, improving outcomes and reducing side effects. The rise of personalized medicine also highlights the importance of genetic databases and the need for more comprehensive genetic research. Large-scale genetic studies have provided insights into how genetic variation contributes to health and disease. These databases contain information on millions of individuals' genetic sequences, along with data on their health outcomes. By analyzing these vast amounts of data, researchers can identify new genetic variants that are linked to specific diseases, which in turn can inform the development of new treatments. One notable example of this is the growing field of pharmacogenomics, which studies how genetic variation affects drug response. By identifying genetic markers that predict how a person will respond to a particular drug, researchers are working to create more precise and effective medications. This could one day make treatments not only more effective but also more affordable by reducing the need for trial and error in prescribing medications. Alongside the excitement of genetic discoveries, however, come important ethical considerations. As our ability to modify and manipulate genetic material improves, so too does the need for thoughtful discussions about the ethical implications of these technologies. The potential for gene editing, for example, raises questions about what is acceptable in terms of altering the human genome. While the ability to edit genes to correct mutations and prevent diseases is incredibly promising, it also opens the door to potential misuse. Concerns have been raised about the possibility of designer babies, where genes are altered not for medical purposes but to enhance certain traits, such as intelligence or physical appearance. This raises important ethical questions about equality, consent, and the societal implications of such interventions. Additionally, gene editing technologies could inadvertently lead to unintended consequences, such as off-target mutations that cause harm. As such, it is crucial to establish ethical guidelines and regulations to ensure that gene editing is used responsibly and for the benefit of society. The ethical considerations surrounding genetics are not limited to gene editing alone. The expansion of genetic testing has also raised concerns about privacy and discrimination. As genetic testing becomes more widespread, there is the potential for individuals to face discrimination based on their genetic makeup. For example, an individual who is genetically predisposed to a certain disease could be denied health insurance or employment opportunities based on that information. To address these concerns, laws such as the Genetic Information Non-Discrimination Act, (GINA), in the United States have been enacted to protect individuals from genetic discrimination. However, as genetic testing becomes more ubiquitous, there will likely be additional challenges in ensuring that individuals' genetic data is protected and used ethically. As we learn more about the vast scale and complexity of our genetic code, the possibilities for improving human health continue to expand. The ability to map, sequence, and manipulate DNA has the potential to revolutionize medicine, agriculture, and countless other fields. Yet, with these advancements come challenges that must be carefully considered. As genetic research continues to unlock the secrets of the human genome, it is essential to maintain a balance between progress and ethics, ensuring that these technologies are used to benefit society while protecting individual rights and freedoms. The ongoing exploration of human DNA promises to reshape our understanding of health, disease, and the very nature of life itself. It is a field where science, ethics, and human experience intersect, and where new discoveries are continually redefining what is possible. As we continue to navigate this complex landscape, one thing is certain: the study of DNA will remain a cornerstone of scientific inquiry for generations to come, as our understanding of DNA continues to evolve. One of the most profound shifts in the field of genetics is the realization that human genetic material is not just a passive blueprint for building an organism but a dynamic system that actively interacts with the environment. This interaction between genes and the environment is at the heart of the burgeoning field of epigenetics, which studies the chemical modifications to DNA and associated proteins that can alter gene expression without changing the underlying genetic code. These modifications can be influenced by a wide range of environmental factors, including diet, stress, toxins, and even social interactions. The concept of epigenetic inheritance, where these changes can sometimes be passed down to future generations, challenges the traditional view that genetic inheritance is solely based on the transmission of DNA sequences. Instead, epigenetics reveals how the environment can shape the way our genes are expressed, influencing everything from our risk for disease to our response to various environmental factors. The growing body of epigenetic research has begun to shed light on how lifestyle choices, such as what we eat and how we manage stress, can impact our long-term health. For example, studies have shown that children born to mothers who experienced high levels of stress during pregnancy may have altered epigenetic marks on their genes, which could affect their susceptibility to mental health disorders later in life. Similarly, the foods we eat can influence the expression of genes related to metabolism and disease prevention. Certain nutrients, such as folate and antioxidants, can promote healthy gene expression, while diets high in processed foods and sugars can contribute to gene changes that increase the risk of diseases like obesity and type 2 diabetes. These findings have profound implications for how we approach public health and disease prevention, emphasizing the importance of not just genetic predisposition but also the environments we create for ourselves and future generations. Epigenetics has also illuminated the role of gene-environment interactions in complex diseases such as cancer. Cancer is not simply caused by a mutation in a single gene but is the result of a combination of genetic mutations and environmental influences that disrupt normal cellular functions. Many of these disruptions are mediated by epigenetic changes that alter the expression of genes involved in cell growth and division. For example, exposure to carcinogens such as tobacco smoke or ultraviolet radiation can lead to the silencing of tumor suppressor genes or the activation of oncogenes, increasing the risk of cancer. Understanding the epigenetic mechanisms involved in cancer development opens up new avenues for prevention and treatment, where interventions could target not only the genetic mutations that drive cancer but also the epigenetic changes that regulate gene expression. One of the most exciting areas of epigenetic research is the potential for epigenetic therapies to treat diseases. Just as drugs can target specific genes or proteins to treat a disease, there is growing interest in developing drugs that target the epigenetic machinery that controls gene expression. These drugs, known as epigenetic drugs, aim to reverse the abnormal epigenetic marks that contribute to diseases such as cancer, neurological disorders, and cardiovascular disease. For example, some cancer treatments are already being developed that use epigenetic modifiers to restore the function of tumor suppressor genes that have been silenced by abnormal methylation patterns. Similarly, in neurodegenerative diseases like Alzheimer's, epigenetic therapies could potentially be used to reactivate genes that are involved in memory and cognitive function, offering new hope for treatments that go beyond traditional approaches. Alongside epigenetics, the field of genetic engineering is also making incredible strides. The advent of tools like CRISPR-Cas9 has revolutionized the ability to precisely edit genes, enabling scientists to correct mutations that cause genetic disorders and even introduce new genetic material into organisms. In the context of human health, genetic engineering holds the promise of curing genetic diseases, such as cystic fibrosis, sickle cell anemia, and muscular dystrophy. These diseases are caused by mutations in single genes, and gene editing could potentially remove the defective portion of the gene and replace it with a healthy one. However, while the potential benefits of gene editing are vast, there are significant ethical and safety concerns that must be addressed. The possibility of unintended genetic consequences, off-target effects, and the ethical implications of modifying the human germline, which could affect future generations, have sparked ongoing debates about how and when gene editing should be used in humans. Genetic engineering also extends to other areas, such as agriculture and biotechnology. Scientists are developing genetically modified crops that can withstand extreme weather conditions, resist pests, and improve nutritional content. These genetically engineered organisms have the potential to help address global food security issues, especially in the face of climate change and a growing population. However, the use of genetically modified organisms remains controversial, with concerns about environmental impact, biodiversity loss, and the ethical implications of altering the genetic makeup of living organisms. As such, there is an ongoing need for robust regulatory frameworks that ensure the safety and ethical use of genetic engineering technologies. The potential applications of genetic research extend far beyond medicine and agriculture. In the field of forensic science, genetic analysis is already being used to solve crimes and identify individuals. DNA profiling, which examines specific regions of the genome, has become a standard technique in criminal investigations, helping to link suspects to crime scenes and exonerate the innocent. Similarly, advances in genetic research have enabled scientists to uncover the ancestry and migration patterns of ancient human populations, shedding light on the history of our species. By sequencing the DNA of ancient remains, researchers can track human migration, understand the spread of diseases, and gain insights into the evolution of human cultures. The information contained in our genetic code is thus not only a map of our personal biology but also a record of the broader story of humanity. As we continue to explore the far-reaching implications of genetic research, it is clear that our understanding of DNA and its role in health, disease, and evolution is only beginning to unfold. Each discovery in genetics opens up new possibilities for improving human life, from curing diseases to enhancing our understanding of the world around us. However, with these advancements comes the need for careful ethical consideration and responsible use of genetic technologies. As we unlock the secrets of our DNA, we must ensure that these powerful tools are used for the greater good, with respect for individual rights and societal well-being. As genetic science continues to progress, the relationship between nature and nurture, the interaction between our genetic inheritance and the environments we create, becomes ever more apparent. The study of genetics, once seen as a purely biological pursuit, now intertwines with fields such as psychology, sociology, and environmental science. The future of genetics will not only be about understanding the individual genes that make us who we are but also about understanding the complex interplay of factors that shape our lives and our world.